fertilization is the union of a sperm and an oocyte wherein the genetic material or chromosomes of both cells that meet will produce a zygote that will later mature into an embryo and then will eventually become a new individual. So there will be a unique characteristic for the individual that will be formed since the genetic materials that it will contain are the combination from the father's genetic material and from the mother. So the process called fertilization occurs in the uterine tubes. So the process of fertilization starts when the sperm travels through the woman's reproductive tract. So it basically starts with ejaculation. So ejaculation, we all know what that is. It is basically when the semen containing millions of sperm cells come into the woman's reproductive tract. So from here, the sperm cells are traveling or transported to the vagina and then to the cervix, then to the body of the uterus, and then to the uterine tube. So the one responsible for the sperm cells motility are the muscular contractions of the uterus, the hormones oxytocin by the female posterior pituitary, and prostaglandin from the semen that stimulate contraction of the uterus as well. So the next phase that we have is called the sperm capacitation. So in this phase, in the process, this is where the sperm are able to release enzymes from the acrosome. So capacitation is when is associated with the removal of adherent seminal plasma protein, reorganization of plasma membrane lipids and proteins. So this reorganization all happens to the one surrounding the head of the sperm. So aside from this, it also causes hypermotility of the sperm that facilitates swimming of the sperm cell to the oocyte. The next phase of the process is called sperm zona pellucida binding. So before discussing further, we must know what is zona pellucida. So zona pellucida is a thick extracellular coat surrounding the oocyte. So the sperm cell will continuously swim until they reach the oocyte or the female egg cell. And then it will bind to the zona pellucida type 3 protein, which has the specificity property that chooses the ligand. So the carbohydrate group in the zona pellucida functions as a sperm receptors, while the sperm is the one called the ligand. In here, the proteins are actually in the cell membranes of the oocytes. So there is a specific protein in the sperm that attaches to the cell membrane. And once it attaches to it, it will open up certain channels in the cell membrane. Next process or next phase of the process is called the acrosome reaction. So in acrosome reaction, we first must know what is acrosome. So acrosomes are part of a sperm cell that contains hydrolytic enzymes, and these hydrolytic enzymes are the one that helps the sperm to penetrate the egg's coat. So in this phase, the acrosome reaction will provide the sperm with an enzymatic drill characteristic to get through the zona pellucida. So the chemicals that the sperm has, for example, acrosin and the proteases, will start to brawl a hole by digesting the pathways through the layer of the zona pellucida. The more the sperm swims deeper through the zona, the more plasma membrane and acrosomal membrane are lost. So acrosomes should not be lost before the sperm encounters the oocyte, because sperm will not be able to bind the zona and fertilization will not occur if this happens. What triggers the acrosomal reaction? So, the one that triggers the acrosomal reaction is once the sperm and the type 3 protein meet. The calcium ions will start binding with the sperm and it will trigger the release of chemicals and causes the hole, hence the fusion of the egg cell and the sperm cell. Next from the process is called the penetration of zona pellucida. So once the sperm cell and one sperm cell and each egg are required to fertilize an egg. So because of the force brought by the flagellating tail of the sperm, along with the enzymes that facilitate the penetration to the zona pellucida, the sperm is able to get inside the oocyte. So the sperm oocyte binding is the next phase of the process. So once the sperm penetrates the zona pellucida, it will bind with the plasma membrane of the oocyte. The sperm will release its genetic materials into the cell, and it will release a haploid cell which contains 23 chromosomes. So the chromosomes are thread-like nucleic acid containing the genetic information in the form of genes. So the egg activation and cortical reaction is one of the phases as well. So in here, the egg activates by completing the second meiotic division and the rise in the intracellular concentration of calcium. So as soon as the male genetic material is present inside the cell, the calcium will tell the cell to finish up meiosis 2, since the oocyte in the fallopian tube is stuck in the metaphase. And after this, the female pronucleus is formed. So it has 23 chromosomes as well. So the zona reaction is one of the process too. And in here, the zona pellucida in this phase is catalyzed by the cortical granules that alter its structure. And this reaction shows the major block in polyspermy. So what is polyspermy? Polyspermy is when the egg has been penetrated and fertilized by more than one sperm. So this major block ensures that only one sperm cell will fertilize one oocyte. So in here, the zona pellucida hardens and the receptors are destroyed. We also have what we call the fast block to polyspermy and the slow block to polyspermy. First, we have the fast block. As soon as the sperm attaches and opens the channels to go into the egg cell, the sodium ions will get in the cell to create a positive charge on the walls of the oocyte that prevents other sperm cells from passing through. And next, we have the slow block to polyspermy. In here, to further block the sperm cell, the smooth endoplasmic reticulum will act once the haploid brought by the sperm enters. The smooth endoplasmic reticulum will produce calcium ions, and the lysosomes inside the cells are activated by calcium to fuse with the egg cell membrane. So it releases hydrolytic enzymes that will break the zona pellucida protein and will harden the cell membrane so that no sperm can penetrate. And then the last one is the post-fertilization events. So in this phase, the sperm fuse with the oocyte. So the nuclear envelope of the sperm disperses and the chromatin loses. So this is for the zygote to mature and to grow for the next 9 months until ready for birth. So the pronucleus are formed, then the microtubules will pull them towards each other to complete the process. Once the two cells fuse, a unique genetic code arises that will determine the gender, the hair color, the eye color, and many other characteristics of the baby. So it will now have the 23 pairs of chromosomes and will become a diploid cell, which is a cell that has 2 copies of each chromosomes. So after this, the cilia in the fallopian tube will sweep the zygote to the uterus to implant in the richer uterine lining. In summary, what happens in the process of fertilization is that first we have the ejaculation of male, the semen with the sperm goes into the vagina, and then it passes through the vaginal canal leading to the fallopian tube where the oocyte is, and then the sperm cell enzymes will penetrate the egg cell as spermatocytes enters the oocyte, and then the egg cell will create a layer to prevent other sperms from entering the egg, since only one is required to fertilize an egg. And the genetic material brought by the sperm and the genetic material will fuse, resulting into having a zygote, and that will later mature into an embryo, leading to growth and maturation of a new individual.
Now let's move on to the implantation. So when the blastocyst continues to fallopian tube and reaches the uterus about 4 or 5 days after fertilization, the outer cells of the blastocyst contact cells of the endometrium lining uterus, and then the blastocyst embeds in the endometrium, and this process of embedding is what we call the implantation. Now, to give you a background of implantation, let's answer this question. So, what is implantation? So, as a nurse, probably when we heard or think about pregnancy, the first thing that came to our mind is the implantation. So, behind the pregnancy, the body has been worked so hard from the moment that the conception occurred, and this is where the implantation starts. Okay, so the implantation is the process in which a developing embryo moving as the blastocyst through the uterus. It makes contact with the uterine wall and remain attached to it. Until birth. And then the lining of the uterus, which is the endometrium, prepares for the developing blastocyst to attach it via internal changes. So the implantation is the beginning of the development of the placenta in embryo as the fertilized eggs burrows into the thickened lining of the uterus to establish blood supply. And the figure attached is the blastocyst. So it consists of outer layer cells, which is the trophoblast, and the inner cell mass, which is the embryoblast. So after the fertilization, they start having the cell division through the process of mitosis. And it begins to divide into 2 cells, then 4 cells, and then more cells as the division continues as the fertilized egg continues to move down to fallopian tube. The fertilized egg is traveled to the uterus and has become a growing cluster of about 100 cells, which also known as blastocysts that I mentioned earlier. And then they move from fallopian to uterus a week after the sperm has fertilized the egg. And then they reach the uterus as blastocysts, and then the blastocyst attach itself to the lining of the uterus, which is the endometrium. So this attachment process is what we called as the implantation. So the implantation is an adaptive process during which the blastocysts establish very close contact with the uterine mucosa, which is the endometrium of the maternal. The implantation begins on the day 7 and it ends on the day 13. The trophoblast cells play key role in this process, wherein they differentiate into cytotrophoblast and syncytotrophoblast, invading the uterine mucosa. And to help you figure out these two, I add these two figures. So as you can see, there's a difference between cytotrophoblast and the syncytotrophoblast. To give you an additional information, placental implantation in humans begin with invasion of the uterine epithelium and underlying stroma by extraembryonic trophoblast cells. The villus cytotrophoblast cells at the tips of some anchoring villi proliferate outwards from the underlying basement membrane to form columns, and from which the individual cells migrate into decidual tissue. And later on, I will give the 3 stages of the decidual tissues. And then these interstitial trophoblast cells invade as far as the superficial layer of the myometrium. And then implantation trophoblast cells invade into the uterine wall, and then the trophoblast differentiates along the 2 main pathway. So the 2 main pathway is the villus and the extravillus. So the villus trophoblast includes the villus tree, which is the bath in maternal blood in intervillus space, while the extravillus trophoblast, or also known as EVT, encompasses all the invading subpopulation of trophoblast EVT cells arise during early development of cytotrophoblast. Okay, so the implantation has 3 stages, and to give you the most expressive explanation, I use 5 stages and explaining the process of implantation. So first is attaching of the blastocyst to the endometrial epithelium. Next is invasion of the trophoblast cells in the endometrium. And then the third one is reparation of implantation defect in the endometrium. And then the stage 4 is the formation of uteroplacental circulation. And the last stage is the manifestation of decidual reaction. So, first one is the attaching of the blastocyst to the endometrial epithelium. It follows the zona pellucidalysis. The blastocyst attaches to the endometrium with embryonic pole, and this pole is by which the embryoblast is placed. And then the invasion of cells in the And then the second stage is the invasion of cells in the endometrium. So let's move on to the next slide. Okay, so for the second stage, we have the invasion of the trophoblast cells in the endometrium. So this stage follows as the result of contact to trophoblasts with the endometrium trophoblast cells and differentiate into two populations like I mentioned earlier. So we have the syncytial trophoblast that invades and erodes quickly and endometrial stroma and the uterine glands. It forms around blastocysts, a large, thick, and multinucleated mass without obvious cell boundaries, and the erosion of the uterine mucosa is caused by proteolytic enzymes that produced by these cells. And then the cytotrophoblast gradually replaces the original trophoblast. The cytotrophoblast cells are mitotic active and form new cells generation. And then migrate into blastocyst cavity where give rise to extraembryonic mesoderm or migrate externally to complete masses of the syncytotrophoblast. And then I add two figure here. As you can see, we have the first one is the development between 5 to 6 days, and then followed by the development within 7 to 8 days. And then the next stage is the reparation of implantation defect and in endometrium. So the stage 3 is the reparation of implantation defect in endometrium. So after the penetration of the blastocyst, the defect in the epithelium is filled with closing plug. And then by day 12, the site of implanted conceptus appears as a minute of elevation of the endometrial surface and covered with regenerated epithelium. And then the stage 4 is the formation of uteroplacental circulation. So around day 9, isolated space cavities called lacunae are formed in the syncytotrophoblast, and they become filled with nutrition secretion from the eroded endometrial glands and with the maternal blood form ruptured maternal capillaries. And between day 11 and day 13, adjacent lacunae fuse to form lacunar network in which the maternal vessel open through which maternal blood flows. Now, for the last stage, we have the manifestation of decidual reaction. So, the conceptus evokes of the interendometrium. It consists of endometrial stromal cells that cell changes that enlarge and accumulate glycogen and lipid droplets. In vascular alteration and granule alteration, uterine glands referred to as the decidual reaction. And when the conceptus implants in the endometrium, the continued secretion of the progesterone causes the endometrial cells. So, now, when the conceptus implants in the endometrium, the continuous secretion of progesterone causes the endometrial cells to swell further and store even more nutrients. And these cells are called as decidual cells. And the total mass of the cells is what we call as the decidua. So the following implantation, the endometrium is known as the decidua and consists of three regions. So we have the decidua ballacis, the decidua capullaris, and the decidua parietalcis. So the first one, the decidua ballacis, lies between the chorion and the stratum ballacis of the uterus, and it becomes maternal part of the placenta. And then the second one is the decidua capullaris that covers the embryo, and it's located between the embryo and the urine cavity. And then the third one is the decidua peristalsis, and it lines the non-involved areas of the entire pregnant uterus. And that's all for the implantation.
So now, I'll be discussing the development of trophoblasts, but before we begin the discussion, let's define the word trophoblast first. So, in Greek word, trophe means nutrition, and blast is the primordial cell, which means it is the precursor cell or the origin that came from the mother and father. So what is the function of trophoblasts? Trophoblasts play an important role in embryo implantation and interaction with the endometrium of the maternal uterus following the decidualization. The trophoblast is composed of two layers. An inner is the cytotrophoblast and an outer is the syncytiotropoblast. So now let's discuss the difference between the cytotrophoblast and the syncytiotropoblast. The cytotrophoblast is trophoblastic stem cells because it can produce new type of cell, and compared to syncytiotropoblast, it is not stem cells because it cannot produce new type of cell. When it comes to nutritional supply, the cytotrophoblast cannot establish nutritional supply between the mother to fetus, and as opposed to the syncytiotropoblast can give nutritional supply between the mother to fetus. Cytotrophoblast is formed by individual cell, and syncytiotropoblast is specialized and it is multinucleated with no individual cell. And as you can see on the picture, the cytotrophoblast is distinct compared to syncytiotropoblast. When it comes to producing hormones, cytotrophoblast layer does not produce any hormones compared to syncytiotropoblast that can secretes progesterone, leptin, HCG, and the HPL. So now let's discuss the trophoblast differentiation. So after the fertilization or the 8th day, the trophoblast separates into two layers, which are the cytotrophoblast and the syncytiotropoblast. So here you can see the cytotrophoblast where the cell remain distinct to each other, and then here you can see the syncytiotropoblast where the cell fused and forming a nucleic mass, and where the cell boundaries are not distinct to each other. The syncytiotropoblast, which is the outer layer of the trophoblast, it secretes the enzyme which then digests the embryos way into uterine lining, and then will establish contact with maternal blood. So it can absorb the nutrients. Then, the syncytial trophoblast will produce hormones such as HCG, progesterone, and leptin.
During the second week of development, the inner cell mass will flatten to become an embryonic disc composed of several layers. The hypoblast columnar cell will eventually compose the endoderm and yolk sac, and cuboidal cells of the epiblast will eventually compose the ectoderm and amnion. The cells of the trophoblast become the extraembryonic membrane known as the chorion. These are embryonic cells, but they will not make a part of the embryo's body. This will form the fetal part of the placenta that exchanges nutrients with maternal blood. And the chorion will suppress the mother's immune system so that the mother's immune system does not attack the embryo. Around day 8, cells of the hypoblast contribute to the formation of the extraembryonic membrane known as the yolk sac. It serves as the formation of the first blood cells, the formation of the germ cells which will ultimately migrate to the gonads after reproduction, and a few other minor functions before it degenerates. 8 days after fertilization, the cells of the epiblast contribute to the formation of an extraembryonic membrane known as the amnion. The amniotic sac will eventually surround the entire embryo and fetus and be filled with amniotic fluid. This fluid originates as a filtrate from maternal blood, but later urine secreted by the fetus will contribute as well. The amniotic fluid prevents the fetus from drying out, prevents tissues from adhering to fetal cells, and will help absorb shock, and it was the development of amnion. Beginning 12 days after development, a space develops between chorion and amnion, yolk sacs and embryo, and it is known as the extraembryonic xylem. In the third week of development, starting day 15, a process known as gastrulation occurs. Cell migration and differentiation produces the 3 fundamental tissues of the embryo. The hypoblast becomes the endoderm. The epiblast becomes the ectoderm, and between those two, it forms a new layer known as the mesoderm. From these embryonic tissues will come the cells to compose the body of the fetus. From the endoderm comes the epithelial lining of the GI tract, the lining of the urinary bladder, gallbladder, and liver, and the epithelial lining of the respiratory system from the pharynx through the lungs, the epithelium of some of the glands such as the thyroid, and of parts the reproductive system. From the tissue known as the ectoderm will develop the entire nervous system, the epidermis of the skin, hair follicles, the lens and cornea, the internal and external ear, the pineal gland, the epithelium of the oral cavity, and other structures. From mesoderm, it develops the skeletal muscle, cardiac muscle, the most smooth muscle, cartilage bone, the dermis of the skin, the middle of the ear, the vascular tonic of the eye, the epithelium of the kidneys, and the epithelium of the gonads.
Men, and especially women, possess various organs that are optimally suited for the creation of another human being. The ovary possesses about 500,000 eggs embedded in follicles. Several follicles, and therefore eggs, mature in each cycle. The dominant follicle grows to about 2 centimeters, and the fimbriated end of the fallopian tube, which eventually catches the egg, moves to the site of ovulation. The egg consists of an outer layer called the corona radiata. Underneath is another protective layer, the zona pellucida. Both layers must be penetrated by the sperm cell in order to reach the cell nucleus that contains 23 different chromosomes. Each chromosome contains DNA. DNA is the blueprint for building our body cells. Eye color, body size, arrangement and function of our organs. It's all in there. If two identical chromosomes occur in one cell, we can later speak of trisomy. The egg cell is transported inside the fallopian tube with the help of cilia and mucus produced by glands in the walls of the fallopian tube. Additional contractions of the muscle layers help to transport the ovum. At a speed of about 3 to 4 millimeters per minute, something else makes its way from the other side of the fallopian tube. Sperm cells, thousands of them, and barely visible. They all have only one goal: reaching the ovum. A sperm cell's nucleus contains the male's genetic information. Through messenger substances emitted by the egg cell, the remaining sperm cells find their way to the ovum. The goal of all sperm cells is to penetrate the two outer layers to reach the chromosomes. The sperm cell penetrates corona radiata and zona pellucida, resulting in the degradation of the acrosome cap. Upon entering the cytoplasm, fertilization occurs, resulting in activation. Activation causes, among other things, a structural change of the zona pellucida so that no further sperm can penetrate. In addition, activation causes the ovum to complete its second maturation division. In this process, the sister chromatids are pulled into opposite poles, and the female pronucleus and a polar body are formed. The sperm's tail and head gradually degenerate into cytoplasm. The condensed sperm nucleus rapidly increases enormously in size, and the chromatin decondenses. Thus, a male pronucleus is formed with 23 unreplicated chromosomes that possess the genetic information of the father. As the two pronuclei approach, both double their DNA, resulting in replicated chromosomes. Finally, syngamy occurs, in which the pronuclei dissolve and the chromosomes of male and female are properly aligned in the spindle apparatus. Now, the cell is called a zygote, which is the very first cell of the new human being. The stage of cleavage begins. In this process, the cell continues to divide. 2 cells, then 4, then 8. At a 16-cell to 32-cell stage embryo, we speak of a murula, since the shape resembles a mulberry. The cells are still enclosed by the zona pellucida. Thus, after each cell division, whilst the number of cells increases, the size of each cell diminishes. Finally, the blastocyst is formed. The blastocyst consists of a fluid-filled cavity, the cells of the embryoblast and the trophoblast. The embryoblast is the initial structure of the new human being, whereas the trophoblast is the origin of placenta and membranes. Let's see the individual stages in the fallopian tube. Fertilization occurs at the end of the tube. The first cell division takes place about a day after fertilization. Eventually, further cell divisions happen forming the murula. And in the uterus, the blastocyst is created. At this stage, the mucous membranes of the uterus or womb has already been prepared for the implantation of the blastocyst. Usually, the blastocyst implants in the posterior wall of the uterus. However, implantation can also occur on the side, as we will see in a moment. The blastocyst floats in the protective and nourishing uterine mucus. For implantation in the uterus, the blastocyst must first hatch from the protective zona pellucida. Then the blastocyst burrows further and further into the uterine lining, like a parasite. Let's take a closer look at this. The endometrium has a protective epithelium and capillaries filled with blood, as well as mucus-producing glands. The trophoblast cells fuse with the endometrium and a syncytial trophoblast is formed that will nourish the embryo. In addition, hypoblast and epiblast arise from the embryoblast. The embryo burrows further into the endometrium, rapidly increasing in cell mass and leaving a wound, which may result in a completely harmless implantation bleeding. Finally, the amniotic cavity is formed. As the process continues, the yolk sac is created, as well as a non-cellular plug that is later sealed by migrating uterine epithelial cells. The trophoblast rapidly increases in size, with the amniotic cavity and yolk sac remaining relatively small. This gives rise to the chorionic cavity and the body stalk. Three germ layers form from the primitive streak and node. This process is called gastrulation. The germ layers ectoderm, mesoderm, and endoderm eventually differentiate into various tissues and organs. In the course of the third week of development, neurulation occurs. At that time, the foundation for the central nervous system is laid. The neural groove is formed, closing from the center to the sides. At the sides, small bumps called somites can be seen. The neural groove becomes the neural tube, which later forms, for example, spinal cord and brain. Those cells later become the neural networks, which enable us to think. In the fourth week of development, this process is almost complete. The embryo is still in the amnion, which is filled with amniotic fluid that has numerous functions. The optic placodes, which are the basis for the inner ear, can be seen, and the heart is already pumping blood through the intra-embryonic blood vessels. The yolk sac and the body stalk continue to nourish the embryo. The chorion is the outer membrane and is connected to the body stalk. The chorionic villi are surrounded by maternal blood. This allows them to carry nutrients to the embryo and nourish it. We'll see more on this later. During the fourth week, 3 pairs of pharyngeal arches appear. In addition, the upper limb buds are visible first, followed by the lower limb buds. The embryo is now curved in a C shape. The caudal eminence is tail-like in shape. The somites, which give rise to the cells forming the skeletal muscles and components of the skin, among other things, can still be identified. The embryo continues to grow rapidly, measuring approximately 4 to 5 millimeters by the end of the fourth week. The lens placodes and olfactory pits are formed. The later arms and hands, as well as legs and feet, can be already well identified. The hands will develop from paddles to separate fingers by the 8th week. The embryo is about 23 millimeters long by the middle of the 8th week. The legs will not lose their webbed feet until a few days after the hands. Embryogenesis turns into fetogenesis in the 9th week, and in the 3rd month, that is, from the 12th week on, the fetus already has a human-like appearance. The eyelids are closed. The skin is translucent. In the following weeks, the fetus grows and the ossification of the fetal skeleton takes place, so that the bones are clearly visible on ultrasound images by the 16th week. In addition, the body is completely covered with downy lanugo hairs, which help, among other things, to keep the vernix secreted by dedicated cells on the skin. The vernix protects the skin from direct contact with the amniotic fluid. From about the 18th week, the mother can feel the baby's movements. In addition, the sucking reflex has already been established. By the 28th week, the hair on the head, eyebrows, and eyelashes continue to grow. In addition, the eyes open. The fetus gains a lot of mass in the last few months, and the lanugo hairs and the vernix gradually fade away. It is also slowly getting tighter and tighter in mom's belly. At first, the fetus is supplied by the body stalk and yolk sac, but then more and more by the umbilical cord and placenta. Since the fetus possesses both the DNA of the mother and of the father, the blood of the fetus must not mix with the blood of the mother. The placenta is responsible for this. It is responsible for the exchange of substances between mother and fetus. The chorionic villi, which were previously completely distributed over the chorion, have regressed to other parts of the uterus. The remaining villi are part of the placenta. Maternal blood, rich in oxygen and nutrients, enters the intervillous space via the arteries. Veins carry away the oxygen and nutrient-poor blood. The chorionic villi extract oxygen and necessary nutrients from the mother's blood. Nutrients include carbohydrates, amino acids, fats, vitamins, and iron. Oxygen and nutrients are then transported to the fetus via the veins with the help of the umbilical cord. However, waste products also reach the villi via the arteries. This allows the fetus to release its waste products, such as carbon dioxide and urea, into the maternal blood. The mother's organism then takes over the excretion of these substances. After about 38 weeks, the fetus is fully developed. It measures 50 centimeters and weighs around 3,000 grams. The birth takes place now or in the next 2 to 4 weeks.
Now that we have discussed the fertilization, implantation, and trophoblast development, we hope you have gained some insight into our report.